Hello, my name is Delaney. This is my presentation, and it aims to introduce the measles virus or the infection of the skin. This virus's taxonomy, structure, pathology, and epidemiology of the disease. This presentation will also cover treatment and prevention. So, what is measles? Measles is an airborne disease that is caused by the Ebola virus and is very contagious. This microbe will cause fever, rash, cough, and red eyes, and it can lead to detrimental problems if the infection is not under control or is contracted by an immunocompromised individual. The measles rash is maculopapular, which usually lasts about 5 to 6 days. It begins near the hairline and moves downward and outward as our virus will proliferate or grow, and it will eventually cover the entire person. This is important to note that this is not the same thing as German measles or rubella. Although similar in symptoms, the virus itself is different. Some nomenclature for measles. This microbe is identified by a few common names. The ones we are most familiar with are Rubella, red measles, or 10-day measles. Its scientific name is measles morbillivirus, according to the International Committee on the Taxonomy of Viruses. Measles is described as early as the 9th century by a Persian physician. I'm not even going to try and pronounce his name. His first name is Abu. He goes by, I'm going to butcher this as well, Rhesus. In 1757, however, Francis Home is the one that demonstrated the infectious agent in blood responsible for carrying the virus. Moving forward to 1846, Peter Panum described the incubation period of measles and its lifelong immunity in a human after recovery from infection. Perhaps one of the bigger discoveries for this disease, however, occurred in the 1950s. John Enders and Thomas Peebles, sorry, isolated the virus in a human kidney tissue cultured in 1954 from a Massachusetts student. The student's name was David Edmonston, and so Enders and Peebles transferred their Edmonston B strain of measles into a vaccine. Here's a timeline that spans from the 9th century. Um, when we see our Persian physician distinguish smallpox from measles, and it goes all the way to 2020. And this is going to just note the important steps in discovering and researching and developing a preventative technique for measles. Highlighted in red are the important phases from the previous slide, as we see the earlier description is when we see measles through blood. We have the incubation and lifelong immunity, the first isolated measles virus, and then it leads towards vaccination methods. Taxonomy, by definition, is the grouping together of biological organisms based on characteristics. Viral taxonomy is based on the various characteristics that can distinguish. See, can I move this? No. That are based on the various characteristics. I'm sorry. Let me start over. Viral taxonomy is based on the various characteristics that can distinguish viruses from one another, including the molecular composition of the genome, structure of the virus's capsid, whether or not it is enveloped, the gene expression program used to produce our virus's proteins, host range, pathogenicity, and sequence similarity. Here's the first part or the first half of the measles virus taxonomy, beginning with ribovira, which is a viral realm that contains all RNA viruses that replicate by means of RNA-dependent RNA polymerase. As we move down, the next lineage is composed of viral organisms made up of RNA, but we continue down, we start to see the negative strand RNA. Then we see the single strand negative stranded RNA viruses. And then the other three lineages start to become more and more complex. As we move down in taxonomy, I don't know what's going on. Okay, there we go. As we move down in taxonomy, we see Paramaxioviridae. This is the lineage of our virus. It is negative strand RNA viruses where the host is generally going to have a vertebrae and it's going to cause a respiratory infection. We move down to morbillivirus. This is our viral lineage where we see infection in humans, dogs, cattle, cats, seals, and cetacea, things like whales and dolphins. These organisms will serve as a natural host to these negative single stranded RNA viruses. Measles morbillivirus is the genus and species name for this virus that causes our measles infection. Here's an image that is going to depict the breakdown phylogenetically of our viral family Paramaxioviridae. We follow it down until we get to morbillivirus. We obviously see measles virus, but canine distemper and rinderpest virus are also present. These two are responsible for infecting the handful of organisms that were mentioned previously. The Ebola virus is approximately 120 to 250 nanometers in diameter, with a genome of single-stranded negative sense RNA, and is closely related to rinderpest and canine distemper, as seen in the slide above. There are two membrane envelope proteins that are important in pathogenesis. The first is called F fusion protein, which is responsible for the fusion of virus and host cell membranes, viral penetration, and hemolysis. The second important membrane envelope protein is the H protein, which is responsible for the binding of the virus to receptors of the host cell. Our Ebola is going to be rapidly inactivated by heat, sunlight, acidic pH, ether, and trypsin. Its spherical enveloped virus has a lipid bilayer envelope coming from the host cell. It is surrounded by a fringe of spikes that are going to be relatively similar in size and length, and probably consist of the complexes of F and H glycoproteins that I mentioned, hence why I mentioned them earlier. These are going to unite to form a biologically active fusion complex. This negative sense ribonucleoprotein complex is the basic unit of infectivity because both the protein and the RNA are required for the successful propagation of the virus. This viron is 15,894 nucleotides in length and possesses a helical structure under an electron scanning microscope. Pathogenesis, one of the fun parts of this lesson. Measles is a systemic infection, meaning it infects the entire body. However, the primary site of infection is going to be our alveolar macrophages, which are innate immune cells in the lung, or dendritic cells, which are immune cells that link the innate and adaptive immune systems together. Alveolar macrophages are the first to encounter incoming pathogens and pollutants, and they help orchestrate the initiation and resolution of the immune response in our lungs. 2 to 3 days after replication in the lung, the Ebola virus spreads to regional lymphoid tissues, followed by the systemic infection, once again meaning all over the body. Further viral replication in regional and distal reticulonucleoendothelial sites is a major site of liposome accumulation. 5 to 7 days after infection, during this phase, our infected lymphocytes and dendritic cells are going to migrate into the subepithelial cell layer and transmit measles to the epithelial cells. The requirement for specific cell entry receptors restricts the cell types that the virus can enter during the initial stages of infection in our human host. This is going to highlight the importance to the role of host proteins that are able to support viral replication and modify the cellular environment. This is due to promote viral replication and host proteins that will respond and attack the virus upon entry. Here we see a life cycle or an image that depicts the life cycle of measles in the human body. You can see in the right lung, I believe, the alveolar macrophages and dendritic cells being infected with the virus, slowly spreading via lymph nodes and blood throughout the body, causing infection to be seen on the entirety of our patient. You can see the skin rash starting to form on the arm. Our measles virus is going to be inhaled. You see it coming down the trachea, infecting the alveolar macrophages, moving towards the lymph node, through the blood, the spleen tissue, liver, and essentially it's going to just occur all throughout the body. And so that is why our measles rash tends to occur head to toe. Epidemiology, this is going to occur worldwide. It is heavily seen, or transmission rates, are higher in our underdeveloped countries due to a lack of vaccine development or vaccine implementation program. We will see this come up later as well. This term reservoir, measles is a disease that exists in humans. So there is no known animal reservoir, meaning this disease is not going to be held in animals. You can't get it from another animal. Nor has an asymptomatic carrier state been documented. This means that Rubella will not spread due to contact with an animal that is not a human, or that a variant of the virus can exist in another animal and be dormant. It must be transmitted by humans, is essentially all that is saying. Temporal pattern looks at temperature and season. So measles is going to primarily occur in late winter and early spring in our temperate climates due to the change in temperature and pressure and things like that. So measles is one of the most contagious diseases that we know of. It is spread through airborne routes from respiratory secretions, as mentioned many times. Many cases today occur in children under the age of 1 due to maternal antibodies being weakened in response to our developed vaccine. Beforehand, measles was not seen in infants because of the maternal antibodies. They were stronger when we had less vaccine and therefore passed down from mom to baby, so our baby was able to fight off this infection easier. What used to be around 1 million annual deaths worldwide has dropped to approximately 146,000 deaths in 2013, and these numbers were reported to the World Health Organization. Come the year 2000, however, measles was no longer endemic in the United States. But if we jump to 2008, there was an increase in measles that is related to European travel, meaning people in the United States were coming in contact with people in Europe, or Europeans were coming to the United States and coming in contact with us. Individuals who were not vaccinated contracted measles and then spread it to other individuals around them that were not vaccinated. Today, many epidemics are popping up across the United States due to this unvaccinated or too young to be vaccinated. These kids or these students going to school. So in 2018/19, this study reviewed cases of measles outbreak among partially vaccinated and unvaccinated children and adults. They reviewed electronic literature from 1979 to 2019, and the papers were studied on the basis of the following words: measles, vaccine hesitance, child vaccination, vaccine refusal, public health, and prevalence of outbreak, in order to possibly link the recent spread of measles in the US and Canada to a lack of vaccination. The review states that there is an indicated risk of occurrence for measles that correlates with an elevated risk in unvaccinated individuals. More specifically, the risks will increase for children 18 years old and below due to contact with other children who may not be vaccinated. Measles infection among the unvaccinated individuals is associated with the majority of measles outbreaks in the United States and Canada. Here is a table from this paper that is going to show the increase in cases just from 2018 to 2019, just 1 year's difference. The United States had practically 0 cases all of 2018 until we hit December, when we see 372 cases start to pop up. We see this large spike of infections continue throughout 2019, and it starts to die off again towards July, August, and September. A similar trend is seen in Canada. However, the reported cases are far fewer than ours, but the trend is still relevant. We start to see an increase in cases from 2018 to 2019. The same study looked at specific areas in the United States that had a large number of cases reported and compared it to the number of individuals in that area who were infected to the vaccine rate of that same area. This specific example in the chart shows the number of cases in Washington State based on age, and at the bottom of the chart shows the number of individuals that were vaccinated against measles. So this shows a clear representation or relationship between vaccine administration and measles infection rate. We see that 52 and 17 cases are responsible from ages 1 to 18 years old, and 62 of our case numbers are in individuals who had 0 doses of vaccine. Um, we start to see that number get less and less as we get more dose of vaccine. That 11 is a little hidden by the screencastify thing. But the 11 under vaccination status is for unknown vaccination status. So this chart kind of proves that we see our vaccination status has an impact on the number of cases that we are going to see in a specific area. The study has similar findings for other areas of the country like New Jersey, California, and New York. The Ebola virus does not have an antiviral cure or a treatment, so prevention and supportive care is very, very important. Immunoglobulin is used as supportive care if the patient is unvaccinated for measles, and then the focus is going to shift towards treating our symptoms like fever, rash, and dehydration. In 1963, an inactivated and a live measles vaccine was licensed for use in the United States. The inactive vaccine, however, was withdrawn in 1967 because it didn't really protect against measles very well. Each of the varying strains of living attenuated measles vaccine was withdrawn, and now the vaccine is delivered as a trio with mumps and rubella, the German measles that I mentioned at the beginning of this slideshow. The measles vaccine administered with MMR has pretty much eliminated measles in the United States. However, due to the infectious nature, we cannot rely on herd immunity. Therefore, vaccination is the best method of control. Before vaccination, almost everyone got measles, but due to our successful creation and implementation of vaccination, measles was eliminated in the United States in the year 2000. Today, there are outbreaks due to a lack of vaccination. Oftentimes, this is seen in small communities of under-immunized individuals, accounting for most of the cases for our many outbreaks. So here are my sources for the research and for the photos. I hope you enjoyed my presentation and you at least learned something new about our measles disease. Thank you for your time and thank you for listening. Have a blessed day.